Good evening, everyone. My name is Minty Ledger, and I'm the Interim Executive Director at Cambridge University Health Partners. It's fantastic to have you here with us this evening, and I'd like to welcome you to the second Cambridge Biomedical Campus on Virtual Tour event. Over the next few months, this series will bring our clinicians, researchers and academics direct to your laptops, tablets and phones as they explain their work, the challenges they face and their thoughts on what the future of healthcare will look like. Tonight, I'm really pleased to introduce Professor Rebecca Fitzgerald, who is going to talk to us about her practice-changing research on the early detection of cancer. Rebecca is the Professor of Cancer Prevention at the University of Cambridge, where she directs a multidisciplinary research programme on the early detection of cancer. She also practices medicine as an honorary consultant in gastroenterology at Addenbrooke's Hospital. The focus of her research group, located in the Hutchinson MRC Research Centre, is on understanding how cancer of the esophagus develops and how we can apply this knowledge to its early detection and treatment. Tonight, Rebecca will explain how she has developed the Cytosponge, or sponge on a string pill test, as a screening tool to identify people at greatest risk of cancer of the esophagus, all the way from the initial idea through to the latest clinical trial results. Throughout this evening's talk, you can ask questions at any time using the Q&A box, which will then be addressed at the end of the presentation by Rebecca. To reassure you all, you won't be seen or heard during this event, but it will be recorded and made available on our website afterwards, and we'll also send you a link to the recording by email. Many thanks indeed to Rebecca for her time this evening, and also to Jill Wilson and Tony Taylorson, who've been working very hard behind the scenes to bring you this event tonight. So with no further ado, let me hand over to Rebecca, and I do hope you enjoy your evening with us. Thanks.
Thank you very much for the invitation to talk this evening on catching cancer early. And I've subtitled this talk, How Long is a Piece of String? And I'm going to come back to that at the end. So I should first just say that I have got a disclosure to make because I'm named on patents related to the Cytosponge, which has been licensed to Covidien, now Medtronic, and that's going to be the subject of my talk today. So first of all, let's just take a step back and really think, is it even scientifically plausible to think about early detection of cancer? How long does a cancer actually take to develop? And on this cartoon, you can see that I've started off with, on the left-hand side, with a group of cells, one of which has a mutation. That's just a mutation in one single cell. And the host, not surprisingly, wouldn't know anything about the fact that that was happening. And that's a very common occurrence, especially with aging. And sometimes those cells can gradually start to expand, and you may develop a pre-malignant condition. We might call that dysplasia or in situ carcinoma. Still, this is very tiny. The host will not have any symptoms and be oblivious to it. And gradually that will start, in some cases, to become a locally invasive cancer until it may go all the way to a really lethal metastatic cancer. So how long do you think that process takes? Well, actually, we understand increasingly that it takes up to many years, often more than 10 years. This is a very gradual evolutionary process. And that means because it's so slow, we've got the opportunity to find these lesions early and to intervene, ideally before the individual has got symptoms, because as you see on the bottom there, the symptoms really don't occur until quite far along that whole process. But as we're going to explore in this talk, how early do you want to detect it? How are you going to be sure that this will actually be a significant condition for the patient? We don't want to be just finding things which are insignificant to the person that could just stay indolent or dormant. So there's an opportunity, because it's so slow to develop, but also a challenge to ensure that we really find those clinically significant lesions. And then let's just think about this on a societal level. So given that scientifically it's plausible to find these cancers early, how much difference would early diagnosis actually make? These are some statistics from Cancer Research UK, and they've looked at the 10-year survival for the 8 common types of cancer combined. Obviously, each cancer may be a little bit different, but these statistics generally hold true, that if we diagnose a cancer early, we call that stage 1 or stage 2, then around 80% of those individuals will survive for 10 years. On the other hand, when it's diagnosed late, stage 3 or stage 4, and I'm afraid that's the majority of patients that we see with cancer, then the 10-year survival is around 26%. So this isn't surprising probably, but a very stark difference in survival. And that's why we're so interested in trying to find better ways of diagnosing cancer early. And this has really become now even policy, and government's life science industrial strategy have set this really ambitious and laudable target to increase our early stage diagnosis from 50% of cases, which is about where it is now, to 75% of cases by 2028. And of course, 2028 isn't that far off now. So, how can we fill that gap and get to 75% of cases? Those statistics perhaps don't mean very much. How does that actually translate to number of new cases? I don't know if any of you follow the Radio 4 Today puzzle for the day. This is one I set for World Cancer Day in 4th of February this year, and really looked at the number of cases that increasing this early stage of diagnosis would translate into. So at the moment, there are about 363,000 of new cases of cancer diagnosed every year in the UK. We know that 81% of those who are diagnosed early survive their cancer for at least 5 years, as I've just told you, compared to 26% diagnosed late. And the question is, how many more people will survive their cancer if we meet the new government target of 50 to 75%? So I've done the maths for you, you'll be pleased to know. The number of people who would be diagnosed is 25% times 363,000, that's over 90,000. Of course, some of those would have survived anyway. So the additional number that will survive their cancer for 5 years is 49,913 more patients. So nearly 50,000 more patients each year would survive their cancer if we were successful in this early diagnosis target that the government has set. So really that would translate into many more lives. But how do we do early detection of cancer successfully? It's easy to flippantly say, well, we'll increase it from 50 to 70%, but how would we get there? And there are a number of different issues to consider. I guess the obvious one might be around the technology. So having new technologies that can do early diagnosis better, that are more accurate, and that are scalable. It's not just about the technology though, because we can have the most fantastic technology, but if the people at risk don't present themselves for testing, then of course it won't do them any good. And one of the big issues in screening and healthcare in general is really reaching the higher risk groups. And so high risk groups for cancer include some of our most vulnerable members of society. And how do we reach those people with these sorts of technologies? And of course, there are a range of public preferences as well around screening and diagnosis. And I'll touch on this again, but there are issues as well as successful diagnosis. Sometimes in screening we have overdiagnosis and sometimes therefore overtreatment. And then of course we do have to consider health economics. How do we pay for this kind of strategy? Will it cost more money or will it save money? So let's think about the technologies. So there are a lot of very exciting developments in technology at the moment. And one of those exciting technologies is to detect cancer in the blood. And this is possible because as cancers develop, they actually shed DNA into the bloodstream. And those fragments of DNA contain mutations in them which are specific to the tumor. And so you can detect them and find out whether a person has got a cancer or not, and ideally where that cancer has come from. So blood cancer tests are being thought of as a pan-cancer test. You don't need to know which particular cancer you're looking for, but the ideal would be that you can detect sensitively whether there's a cancer and where it has come from. This would probably be a two-step test, so you would have the blood test, and then if there was a signal, you would have to have some sort of further test, probably imaging-based, to find out where the cancer lies. And there are huge strides being made with this technology, which is becoming increasingly sensitive and specific, and it's often combined as well as measuring DNA, proteins as well that are shed in the blood that increase the specificity of the signal. You may have heard of breath biopsy or breath testing, and there are some fantastic anecdotes of dogs who have noticed something different about their owner and sort of alerted them to the fact that they may be ill or have a cancer. And through analysing the chemicals in the breath, we can effectively do what the dog does when it sniffs the cancer in a more chemically designed manner by using a mask like this, which you breathe into, and it collects your breath. You breathe into it for about 10 minutes or so, and then the analytes will be collected on these small tubes, which can be analysed by mass spectrometry in the laboratory to again try and get a signal for whether there's a cancer there and where it might have come from. You might wonder, is this only good for lung cancer? But actually it works much more for a whole range of cancers because of the way that we metabolize things through the liver, and these then come on the breath. So this is work underway in a number of different laboratories, but including this spin out from Alstone, which is a Cambridge University spin out, and we're testing this breathalyzer in a trial at Addenbrooke's at the moment. Here's a test you might be more familiar with, because of course we do screen for colon cancer, and these tests have been evolving. The sort of standard test for this is looking for tiny traces of blood in the stool. But one very topical question about screening for cancer is, do you test everybody, and do you test them at the same age? Or can you take into account other risk factors which might distinguish between those people who really need the screening test from some people who are actually probably at such low risk that they may not need the test at all or may not need it so often? And so this is an example of a risk estimate that's being used now in colon cancer, which understands an individual risk more precisely, so that you just don't have one size fits all where everyone gets sent a colon cancer test in the post at the same age, but based on their age, sex, ethnicity, BMI, smoking status, medical and family history, you can get an idea of an individual risk and then whether they need this test or not. And this Q cancer calculator is quite simple to use, and the GP or the individual can do it at their leisure at their computer. So that's by way of background of some different considerations for early diagnosis tests. And what I'm going to do now is focus on cancer of the esophagus or the gullet, which is the cancer that I've been studying particularly. There are different diseases that can occur in the esophagus, and actually two main types of cancer. Squamous cell cancer tends to occur in the top portion of the esophagus, and this is globally the most common, and particularly common in certain parts of the world along the Caspian Sea, Iran, Turkmenistan, and in Asia. We've been seeing rather less of this cancer recently in the UK, in North America and Australia, and instead we've been seeing much more adenocarcinoma, which occurs near to the junction with the stomach. And this is a cancer which tends to occur in people who have chronic heartburn or reflux of acid and bile coming up from the stomach and damaging the lining of the esophagus. And it's this cancer that I've been particularly focusing on. So most patients with cancer of the esophagus unfortunately do present when their disease is at quite an advanced stage, often when they've got difficulty swallowing and are losing weight. So why is it that we don't see them until that advanced stage? Why don't we see more people much earlier? I said that the main risk factor is heartburn, so perhaps we could refer people more routinely for an endoscopy if they have a history of heartburn. But you can start to imagine if we were to refer everybody with a bit of heartburn, which is an incredibly common symptom for an endoscopy, that would be really unmanageable, not feasible in our NHS. So we need to think about an alternative method. We need to try and think of something much simpler that could be done at the GP surgery. And that's the thinking behind the Cytosponge TFF3 test. And this cartoon just compares and contrasts the Cytosponge TFF3 test with an endoscopy. Some of you may have had an endoscopy. It's a very straightforward test, pretty easy to do. Nevertheless, you have to come to the hospital. You're usually sedated. You need several healthcare professionals to administer the test, someone to deliver the endoscope, someone to assist with the biopsies, and someone looking after the airway. You get beautiful views, especially with the modern cameras, down the esophagus, and then you can take tiny biopsies through this forceps in the side channel. So that's the current gold standard. The Cytosponge TFF3 is altogether much more simple. We don't get an image, but it really relies on the fact that you collect a very rich cell sample from a long passage of the esophagus for analysis. And there's so much information you can get from analysing the cells, you don't need to visualise it. So the patient swallows the capsule, which is tethered to a string. The capsule will dissolve when it reaches the stomach, and out will pop this mesh sponge, and then a nurse will pull it back over just a few seconds, collecting cells all the way along the esophagus as it's pulled out. I thought you would just like to see this picture of endoscopy during the COVID pandemic, because endoscopy is an aerosol generating procedure that's caused us really a great deal of issues during COVID-19. And you can see here, this just gives you an idea of the number of people helping in the endoscopy. This is standard for all endoscopy procedures, but also the protective equipment that they're having to wear. And during the pandemic, we have been using the Cytosponge in Addenbrooke's as a more straightforward alternative to test some of our patients when endoscopy restrictions have been really severe. And here's a picture during the COVID pandemic of one of my clinical fellows who has also got some protective gear on, just giving a Cytosponge to the patient, and you can just see how much easier it is in the office setting. So here's a short video just to give you an idea of how the test works. So this is the capsule on the string reaching down the esophagus into the stomach. And in that moist, warm environment, the capsule will dissolve over just a few minutes. And you can see the sponge now being released. And then the nurse will withdraw the Cytosponge, collecting cells from the top of the stomach along the esophagus and grabbing these groups of cells in this sort of mesh sponge as it's pulled out. And what we're looking for is evidence of pre-cancer, particularly, can detect cancer too, but what we're really after is there a pre-cancer and early cancer there? And this is the kind of sample that you get. So with endoscopy, we do a small biopsy, and these are the appearance of the biopsy looking down the microscope. On the right-hand side, you can see the sort of specimen we get from the Cytosponge with these clusters of cells, which are the pre-cancerous cells. So you could just analyze those down the microscope and wait for a pathologist to really identify them as precancerous. But we've developed this lab test called TFF3, that stands for trefoil factor 3, which is a protein. And the antibody to this protein stains the cells brown, so that they're really easy to spot and highlight within this quite large sample. So these are all normal cells, and then these precancerous cells that the pathologist can spot straight away as being trefoil factor 3 positive. And indeed, the stain is so good that we can use artificial intelligence to hone in straight away and pick out these cells for the pathologist to confirm, so that they don't have to screen the whole sample. So we've made this a kind of semi-automated test now from the pathology point of view. You might say, why don't we automate it entirely? And that's because there's a lot of regulation, quite rightly, around automated analysis. And if you can use it as triage assisted and still get a pathologist to sign off, then that is preferable. So what does the evidence show about Cytosponge? Does it work? How good is it? Do people find it acceptable? So we've done a number of trials now, and the first one we published 10 years ago, 2010, just showing how long it really takes to develop this sort of technology. And the first trial was really to see, is this feasible and acceptable in primary care, and try to get an idea of the accuracy. So this was done in East Anglia. And then we moved on to a second trial called the BEST2 trial, and here we were really interested in how accurate the test is. And we were comparing its performance in patients with known Barrett's esophagus, the precancerous condition, to patients without the precancerous condition who just had heartburn but nothing wrong with their esophagus. And those two trials were very successful, and so we moved on to a randomized trial to try and get definitive evidence about whether the Cytosponge would diagnose more cases of early cancer than what the GP usually does. So in this BEST3 trial, which we've just completed in the past year or so, more than 13,000 patients were randomized. I say patients, they were people in the GP surgery, and they were people who have a history of heartburn and who were on medication for that heartburn. And we put the individuals into two groups, half in the standard of care, which means the GPs continued with medication as required for heartburn, and they would refer them if they felt it was necessary. So as they normally would, if the symptoms were getting worse or not responding to the medication, or if they were concerned. The other half were all offered the Cytosponge. They continued to take their medication if required for heartburn and reflux. And the main question we wanted to ask was simply, did we find more cases of the pre-malignant Barrett's esophagus when you offer the Cytosponge compared to standard of care? And these are the main results. So in the usual care arm, we diagnosed 13 cases of Barrett's esophagus. Whereas in those offered the Cytosponge, there were 140. Early on in the trial, the coordinator asked me what I was expecting the ratio to be, and I said about 3 to 1 I was hoping. And I was, as a principal investigator, was blinded to these results. I was not able to be aware of the results until after the statisticians had finished their analysis, and I was really quite flabbergasted by the difference. So this is we convert this to a rate ratio, which accounts for the clustering of the design, which gives 10.6. So a very statistically significant result. Now you might ask, it's all very well to diagnose Barrett's, but did you find more cases of cancer? I'm just going to go through this stepwise. If we compare the proportion of cases with cancer in the usual care arm, we did find there were 3 cases of cancer diagnosed. Unfortunately, they were all quite advanced. 1 died, unfortunately, during the course of the trial. 1 is still receiving palliative care, and 1 had chemotherapy and surgery. I've divided the Cytosponge patients into 2 because they didn't all accept the offer. They all offered the Cytosponge by letter. And in the end, 39% expressed an interest, and 24% of patients actually had the Cytosponge. There were a lot of patients in this arm who were offered it, but didn't take it up. And we all know what it's like. If you get a letter in the post asking you to take part in the research trial, you might not do it. So what about the cancer cases in those arms? So in the cases that were offered the sponge, there were 2 advanced cancer cases, stage 4, and there was 1 earlier stage cancer. And it was very interesting because this patient had expressed an interest in Cytosponge, but prior to actually having it, because of the symptoms, they were referred for an endoscopy. So probably being told about the Cytosponge did alert them that they maybe needed something looking at. And here are the Cytosponge patients. So 123 with Barrett's, and then several with pre-cancer called dysplasia who required treatment, but that treatment was really straightforward, could be performed down the endoscope. 4 stage 1 cancers and no advanced cancers. So what was really striking was the earlier stage of diagnosis when the Cytosponge was offered compared to usual care. This is one of our patients who took part in the trial, Liz Chipchase. And Liz is a retired scientist, lives in Cambridge, and she had a long history of heartburn and hadn't had an endoscopy. And she really took part because she was intrigued about the test. She had a positive Cytosponge test. Here you can see the positive cells staining for that TFF3. And when we did the endoscopy, we were quite concerned about a very large ulcer within a pre-malignant Barrett's. She had an ultrasound down the endoscope, which confirmed that it looked like there was an early cancer there. We removed the area down the endoscope, and she was really fortunate because there was a superficial cancer there, but it was just superficial to all be removed by this resection, just on the cusp of going deeper, and she managed to avoid surgery, and she's doing very well.
Called the Cytosponge, you swallow the device and it helps to find something called Barrett's esophagus, which is a condition that can potentially develop into cancer. Liz is one of those lives it saved. She had no idea she was ill, but volunteered to try the device out of scientific curiosity.
I was just interested in the trial. I'd never heard of Barrett's esophagus, which is what this is looking for. and so I thought, yeah, I'd be interested in taking part. And I was very lucky, because if I hadn't, I wouldn't be sitting here now feeling perfectly healthy.
The idea is simple enough. Once in your stomach, the casing dissolves, leaving this wiry-looking sponge behind. After 7.5 minutes, the nurse pulls it back out, and it scrapes millions of cells from the lining of your esophagus or food pipe.
And I don't know if you noticed, but Liz was chatting there with the string coming out of her mouth while the cytosponge was down. So she tolerated it really very well. And just to go through these different treatments, depending on the stage, when it's early, we can treat it as an outpatient. When it's more advanced, then we're talking about chemotherapy and surgery and 3 to 6 months of treatment. And then often when it's advanced, we focus on trying to improve the nutrition, but with very poor outcomes. So diagnosing early for this cancer is really transformative. As I said at the beginning, it's not just about having clever new tests, it's also about how you get those tests to the people that need them most. So in our trial, 24% in the end had the Cytosponge when they were offered it. That was in the context of research. But one thing we're really interested in now is what the uptake would be if they were offered it by their GP in a more routine setting. And what about all those people that never see their GP that have heartburn? Should we be thinking about maybe offering it in the pharmacy, for example? And there's a lot of interest in self-testing for understanding your susceptibility to various diseases. This is the 23andMe test you can send off for a pack. Is that a good idea or not? We could debate that. I'll be interested to know what you think about it. But you know, if you do a test like this to yourself, then of course you have to be ready for the results, and you might need someone to explain the results to you, and then you may need follow-on tests, and who is going to pick up the tab for that. This is a schematic from Stanford University, where they've been thinking about having sensors in the home that might detect changes in your environment, changes in your appearance, changes in your bowel habits, for example, almost without you knowing it, and highlight if there's an issue. It smacks a bit of Big Brother, perhaps. Maybe that is a good thing. It would be right there without you having to be proactive, or maybe it would be one step too far. These are all interesting things to really think about in the early diagnosis field. Here's an idea of a smart toilet to try and detect bowel cancer. So there's a huge amount of technology being developed, and in the program that I've been leading in Cambridge on early diagnosis, we've been really keen to engage with other expertise across the university, not only in physics and chemistry on the technology side, but also people with expertise in social anthropology, public health, ethics, and law, to really make sure that we're thinking about the implications for society, as well as just on the technology. So where do we see the Cytosponge fitting in the NHS in the future? So primary care is where we focus most of our studies, and particularly thinking about this as a sort of screening application or a triage. So patients who are on medication for heartburn, who might be invited when they have a repeat prescription to have a Cytosponge to see if they need an endoscopy. In secondary care, in a current implementation trial that we're running, this is funded by Innovate UK, we're interested to see if we could use this in patients who've been referred to by the GP for an endoscopy. Could they have a Cytosponge first to help us identify who needs an endoscopy and whether some people could perhaps avoid an endoscopy, particularly at the moment with all the constraints in COVID. But these are very similar but slightly subtly different indications for the Cytosponge. And so we'll need more data to really understand whether there's a role in secondary care as well as primary care. And there's a lot of pressure on the NHS at the moment for rapid and early cancer diagnosis, reduce waiting times, and to prioritise endoscopy at highest risk. So perhaps the Cytosponge could be useful. Just to round this off in terms of our thinking about the different issues, what about the economics? Of course, treating cancer is very expensive. We're getting better and better drugs that are more precise, that are molecularly targeted, but they're also increasingly expensive. And cancer, when it's diagnosed late, costs a lot of money. What about if we could diagnose it early? Would that cost more money or save more money? Some of these economic analyses are quite complicated. This was an estimate from a report in CR UK in 2014, and they reckon that there could be considerable savings to the NHS overall if we diagnose cancer earlier. We have to factor in those economics when we're evaluating these technologies. So this is a sort of food for thought. You know, in the West, we get more and more obsessed with keeping well and our health. But what about globally when many people don't have good access to healthcare, and if we're getting more and more technical? But actually something like the Cytosponge, which is so simple, could also be applicable potentially in some of those low-income countries where we could take it out to rural economies. So this is something we're also very interested in. So I hope I've given you a flavour of some of the research we're doing, some of the complicated issues to consider in early diagnosis, and to come back to my title, how long is a piece of string? And this particular piece of string on a cytosponge is just over 50 centimetres. So I hope that's been interesting. Very happy to answer questions. So I think we'll move to questions now. And of course, I should thank the all of my team members who have contributed to this work, because this is very much a team effort that we have developed over many years. Thank you very much.
Lovely, thank you, Rebecca, for that. That was fascinating. And we've had quite a few questions have come in from not only the question and answer session, but also the chat feature there as well. So we'll just go through them. There may be some that will help inspire more questions, so please feel free to add some more as we go along. So the first one was, how much does the Cytosponge cost in terms of device, staff, lab costs? And is an endoscopy required afterwards to confirm the diagnosis or the stage of progression? How does that work?
Excellent questions. So this has been, the technology in terms of the cost has been licensed to Medtronic, big device company that many of you will be aware of. Medtronic actually make the treatment for pre-cancer of the esophagus as well. So they're a very good partner in this. They haven't fixed their tariffs yet, so it's difficult for me to give you an actual amount. And of course, I'd love this to cost hardly anything, but that's not the case when you then have to make a device that's commercially viable. At the moment, an endoscopy, just to give you an idea, costs around £450 roughly, and the Cytosponge should be certainly less than half of that. And probably once it was starting to be made in, mass produced, could come down further. That would include not only the device itself, but also the lab processing, the antibody test, the pathologist to report it, and getting the result back. But it's still, so I shouldn't really speak for the manufacturer because they're still positioning this. But those are the kind of figures we've done when we've done our health economic analysis with the trial results. The other question then related to that was about whether you have to endoscope if you have a positive test. So yes, and in our BEST3 randomized trial that I showed you, we did offer patients an endoscopy when we found a positive result. And that's how we found the early cancers, for example, the case that I showed you of Liz. So we then go on and do the endoscopy to prove it. What we're very interested in, though, is whether we can do further tests on the cells, which means you could actually get an idea right from that same cytosponge sample, whether there are cancerous cells there. That might mean you don't necessarily, obviously if there's a cancer you want to treat, you need to endoscope them, but there are, all these stages that I was describing to you, and there are patients, when we find they've got Barrett's esophagus, we do monitoring very regularly, and it would be nice if we didn't have to endoscope all of those. So rather than having to endoscope everyone with a positive cytosponge, maybe we could just endoscope those really at the highest risk where we think that they probably are going to need treatment. And that's something we've been doing a lot of work on.
Okay, great, thank you. And one of the things that came up when I was watching the animation of how it happened is it just seemed such a simple but incredibly effective piece of technology. And someone's asked the question about what inspired the idea in the first place. As I say, it seems such a simple but fantastic idea.
Yeah, I mean, it's sort of embarrassingly simple in a way. You know, you spend so long developing something incredibly simple, but that is the beauty of it. And it came about really just from a conversation I was having with my then boss at the time, Professor Michael Farthing in London, and we were just talking about really from a sort of research perspective, some of the issues we were having in getting the sort of samples, the fact you need an endoscopy every time. And he said to me, Rebecca, you know, what you really need is a bottle brush for the esophagus. And that really stuck with me. And when I was then thinking about, you know, some of the issues around late diagnosis and the fact we wanted to push it earlier and the barrier of doing endoscopy, I kept thinking about this bottle brush idea. And when I got to Cambridge around 2001, 2002, I actually made a sort of version of a bottle brush in the engineering lab, in the LMB, in the laboratory molecular biology. They sort of have some, you can get them to prototype things for you. And we made this tiny sort of small version of a bottle brush. I never swallowed it. It's probably a good idea because it might have been quite lethal. And then after that, I started sort of thinking how we could adapt that sort of design. And, you know, back in many years ago, 50, 60 years ago, maybe more, in Japan, particularly where they have a lot of squamous cell cancer, they worked on various balloon type of devices with a syringe that you put a sort of tube down the esophagus and then blew up a balloon to scrape some cells and things like that. And so the idea gradually morphed from a kind of rigid catheter to something that was a soft string, and you could swallow it that way. So it was a gradual evolution of the idea, but trying to keep it as simple as possible. But on the other hand, the lab tests, I mean, the antibody test is quite simple, but looking for cancer, some of those tests, we can get really quite sophisticated with the sorts of analyses you can do, not only looking for proteins, but looking for mutations in the DNA, for example.
Some people have asked a couple of more specific questions about, someone's asked, is there any risk of the string breaking when it's either being swallowed or when it's been pulled back out again?
Yes. So that's what everyone worries about. The string won't snap, but what can happen is the string will pull away from the sponge. Well, if it does, and it has, not very often, less than 1 in 2,000, but it has done, all it means is you have to have an endoscopy, which you might have had to have anyway. Gastroenterologists are quite used to fishing things out of people's stomachs, sadly, because little bits of Lego and things get swallowed from time to time, so it's not a big deal, but it's inconvenient. So yes, it can, the string can pull through the sponge, but not very often, and it's not a big problem.
Okay. Someone has just said that we're talking about the home and self-testing, whether that was viable. They've also asked how long after using the sponge can it be analysed. So if you were having those home tests, if that is how it kind of go, the potential future for it, what would the process be? How is it envisioned?
Yeah. So I don't really see this as a home test, because actually pulling it out, you're better to have someone doing it for you who's a bit experienced. I mean, it's very simple, but actually pulling it out yourself, you have to extend your head a bit, and you know, you might suddenly stop halfway through and get a bit of spasm in your esophagus, and so I think you'd really want someone doing it for you. As soon as it's come out, it can go to the lab to be analysed. So I guess, you know, like any hospital test, it's probably not going to be analysed the minute that it's reached the lab. But the whole processing is pretty straightforward, and in fact we've had a Cytosponge that we were particularly interested in recently for a patient. We had an early cancer. The sample was received in the lab on Friday, and I've got the results, and we've had the weekend in between. So you know, a couple of days. It's not complicated.
Oh, great. Someone's asked, I assume they're asking this on behalf of their child, if it's asking if this cancer is rare or common, and also would it be fine to show this video in school. So I would assume Taddy, I hope that's how you pronounce her name, is talking about the animated video, and I'm guessing that should be okay.
That would be absolutely fine. We'll be very happy for you to show it in your school. In terms of whether it's rare or common, so it's not nearly as common as, say, breast or colon cancer or lung cancer. We see roughly about 7,000 or 8,000 cases per year in the UK. So it's about the 7th or 8th most common cancer in the UK, but it's quite a lethal cancer, so it's more like the 5th or 6th cause of death from cancer in the UK. So it's not super rare, but it's not very common either. But we've seen a lot more of it in the last 20 years for reasons we don't fully understand. But we're seeing 6 times more than we used to see 20 years ago.
So if you're kind of seeing these extra cases, do you feel slightly under pressure to get, well, throughout the last 10 years or so since you said that you've been developing it, do you feel the pressure to get it moving and to get it to hopefully save more lives?
Yeah, I mean, that's one of the real things about doing this kind of research, is that it takes a long time, a frustratingly long time. Some of that is right and proper, because things have to be properly evaluated before you roll them out. And, you know, you want to know it's safe, you want to know it's acceptable, you want to know just how effective it is. And actually, until you do some of these big trials, and that the last one I described to you, where you actually randomize, so you offer some people the test and some people have, you know, don't have the test or have the usual care, you can't really make some of those conclusions. So much as though it would be nice in some ways to kind of, you know, take some shortcuts and get it out there sooner, you also want everything to go through due process. But it has been frustrating at times, and you have to really be patient and persistent. You know, and even now we've done that huge trial, and you can't at the moment go out and just ask your GP for this test. I think we're getting pretty close, but we've still got to go through processes of whether the commissioners think this is ripe, or, you know, what NICE think about it, National Institute for Clinical Excellence, who do a fantastic job of really impartially, objectively appraising the evidence. Because it's all right someone like me saying it's great, but what about someone that is one step removed, that's looking across all the technologies, thinking about the budgets, to really then make those decisions.
Okay, slightly linked to this, and we were always going to, in this time, we were always going to get questions about COVID. Someone has said, why are we still asking questions about cancer after all these years, when potentially, I should say, an effective, safe COVID vaccine is expected in a few months? Again, I'm assuming that there are very complicated reasons why, that you could probably explain far better than I can.
So I'm not quite sure what's behind this question, whether it's to do with
I would kind of think
priorities.
Of it, yeah, well, no, I kind of, the way that I'm reading it would be that science has thrown everything it can at creating a COVID vaccine in a very short period of time. Is the, and sorry for Daniel if I'm taking a guess at what you're saying, why is it that huge amounts of money is being invested in cancer research, and yet we're still asking these sort of still similar questions about how we can detect cancer sooner, treat cancer sooner? But I'm guessing that with research, with the complex natures, there's particular reasons why that is.
Well, I think actually we've made huge progress, and the thing about cancer is it's not one disease, and I think that's something we've really learned much more about. So actually it's lots of different diseases. So, you know, I told you in cancer of the esophagus, there are 2 main types to start with. That's just looking, you know, down a microscope at the pathology. And then if you start looking at the DNA mutations that we get, there are actually many more subtypes than that within those main pathology groups. You know, there are lots of different subtypes that might have had a different cause and may respond to a different therapy. So often I think we've been rather sort of blunt. Our instruments have been quite blunt in the way we've described cancer and just lumped them together. So as soon as you start really then trying to look at ways of detection or cures, you realize that it's rather more complicated than you'd perhaps realized. You know, COVID-19 is actually one virus. But then I think it's quite incredible the progress that is being made, actually. So, you know, I was telling you a bit about the blood test being developed. I was one of those real skeptics that they would really struggle to get anything like the kind of signals that you would need to detect cancers at an early stage. But already in the last 3 years, great strides have been made in that direction. So I think, of course it could be better and there's still a lot to be done, but actually I think we actually have made quite good inroads.
Yeah,
Good optimistic.
Thank you. Then someone's gone on, again, talking about the Cytosponge, this what has been your greatest challenge developing the tool and evaluating it?
Well, that's, how long have you got?
14 minutes.
Lots of challenges on the way, actually. Things that I never envisaged, and it's been a great, and it continues to be a really interesting journey. And one of the really fascinating things that I really enjoyed has been the very multidisciplinary nature of this whole project. So I've learned a lot about knots and strings and material sciences, manufacturing. I've learned a lot about public health and primary care, and how you position this in the clinical pathway, about the difference between screening and diagnosis, about health economics, about the laboratory tests, working with protein antibody tests and DNA tests. So there have been lots of different facets to cover, and that has been challenging at times because obviously you don't know, you can't know everything about everything. So the great thing is you collaborate and you learn as you go. But there are some things, I guess, if I'd have known earlier on, we might have done it a little bit differently. And the regulation side of it has been very interesting. So devices like drugs are very carefully regulated. So that's been a learning curve. In terms of what's been the most challenging thing, I think I knew very little about commercializing technologies, and that has been challenging. And then you get to a certain point when you develop something like this. I'm still doing sort of R&D, if you like, on making the test even better, but you have to hand it over and you have to trust other people. It's not my trials anymore, and other people have to decide if they think it's fit for clinical use. And the commissioners have to make those decisions, and that's absolutely out of my hands. And as someone that's been controlling this for a very long time and doing all these tests, that's quite difficult.
Do you think, because of the kind of nature of how Cambridge operates, and the kind of the unique situation that we have a biomedical campus with 2 world-class hospitals, and you've got the university and you've got other people as part of the Cambridge cluster. Do you think you could have done this anywhere else other than Cambridge, or do you think that the ethos has helped you to develop it?
I'm sure it's helped me. I mean, I haven't had to look very far at all to get all the expertise I've needed. And for example, I met people in the material sciences department. It just took me, a bit of time on the website looking up who was who, and then getting on the email and saying, oh yes, come across. We'll have a look at that material for you. We'll do some micro CT of it, and getting on my bike and going across and meeting these people who were incredibly helpful. And so the environment is fabulous. I'm sure there are other fabulous environments around as well, but I'm a Cambridge graduate. I went away for a while and came back, and certainly I think it's a superb environment.
Great, thank you. Other people have a couple of these sorts of questions. Are there any other uses for this technology that you've developed? Does it have the capability of detecting any other cancers, or actually any other disease that's in this?
That's a good question, because you obviously get a number of different diseases in the esophagus. So yes, already we detect infections of the esophagus. Candida is quite common in the esophagus, for example, particularly with people using steroid inhalers. We've detected Helicobacter pylori, which is the bacteria that can colonize the stomach, which actually can be a risk factor for stomach cancer. So there's those kind of things. Then there's something called eosinophilic esophagitis, which is a sort of allergic condition of the esophagus. Can go along with people who've got other allergic conditions like asthma that can cause swallowing difficulties. And we've detected that, and that's treated by different things, but particularly by exclusion diets and altering your diet. And so there a colleague of mine, particularly in the Mayo Clinic in the US, who has patients who have to have very regular endoscopy because of their eosinophilic esophagitis, and he's been using the Cytosponge to monitor them, so that's been quite interesting. The thing that I'm spending quite a bit of time looking at now is how we could use it to diagnose the other main type of cancer, the esophagus squamous cell cancer, which, as I said, is globally the most common type of cancer, and particularly high rates in Asia, but also along the whole Caspian Sea, and also in other parts of Europe, in Poland. And we're collaborating at the moment with some Polish clinicians to see if we can detect that kind of cancer. So there it's the same device, but you need modifications in what you look for in the lab. So the TFF3 antibody is irrelevant for squamous cell cancer, so you have to use different biomarkers, as we call them, to identify the cells that you're interested in. So I think that's something I'm very keen that we try and focus on, because globally that's a huge burden of disease. Some parts of Africa, I should say, too, and we recently did a small pilot study in Tanzania. But then a very interesting discussion with the researchers there, because they don't really have the treatments they need. So, you know, I then felt quite troubled to think we could go finding these cases if we couldn't offer the treatment that the patient needs. So I think, you know, if you're working on diagnostics, you do of course need to make sure you've got the treatment for the patients as well.
Yeah, of course. I mean, the interest you must have received from this, from you've kind of touched on it, from different areas of the world and different areas of science. How has that affected your ability to actually do to develop it further? Do you have to make choices about which direction you take the Cytosponge in? Because it must be a difficult thing to do.
So yeah, and this kind of research is very applied, and you know, sometimes in Cambridge labs, big universities, you know, you perhaps think, is this really science? You know, you're developing a sponge on a string, you've got the lab test, but, you know, and then I spend a lot of my time sorting out quite mundane things, but very important things about implementing a technology and how we position it in the clinic, and making sure we really have all the evidence we need for NICE and the health economics. So some of that stuff isn't very always as scientifically interesting. Yeah, and as I said, it's multifaceted, because you've got so many different sorts of professionals you're engaging with this sort of technology. So that makes it challenging. But I think, you know, my perspective on this is, having set off on this journey, I'm really keen that you know, we see it through, and I think the evidence suggests that it could be really useful for improving health. And, you know, I see the bad end of this disease when I look after patients with cancer of the esophagus. I chair the weekly meeting where we discuss all the new cases presenting in the region with this cancer and work out how to treat them. And that's really tough. And so, you know, I'm really committed to it doing what we set out for it to do. But that is quite a long path.
One of the kind of one of the questions that's come in is, how have you funded this development, and what advice would you give to potential innovators about how to take their ideas forward? I guess that's always been a challenge for people, no matter what product or device that they have, is how do you go to the market? How do you get that support?
Yeah, I mean, someone said, what's the biggest challenge? And that's probably been one of the biggest challenge. I've been very fortunate with the funding. So funded by the Medical Research Council for all the initial work we did on this, and then funded for the trials through Cancer Research UK, infrastructure support for the clinical research facilities and endoscopy facilities by NIHR. So really excellent funders. And I think one of the things I struggled with was really knowing how to commercialize something. And, you know, we thought about setting up a company initially, and versus license deal, had different sorts of advice, sometimes conflicting, had difficult choices to make. And And in the end, actually, this is an interesting technology because it's both a device and a lab test, and there are very few companies that really do both. So in the end, we've licensed it to Medtronic through a devices company, and we've actually now made a startup company to, amongst other things, it's no detection company, but to help with the lab side, because really it's very different sorts of expertise. So that's been one of the other commercial challenges. And, you know, for some academics, it can be a bit scary to go down the commercial route, or they might also think that it isn't a good thing to do. But actually, if you want to get these technologies to the patients, you have to commercialize it. So I think that's something I feel I've sort of learnt on the hoof. And I guess Cambridge, I think, not alone here, is getting much better at how we really work with academics to help make sure that we do protect the IP and commercialize things where they can be helpful to patients. And that's really important to do. I think that's, you know, something we need to encourage and help our academics with more.
What's next for the Cytosponge? Someone's asked, is it widespread trialing in the primary and secondary care? What is the next stage?
So at the moment, we're doing an implementation trial funded by Innovate UK. And there's been actually some accelerated interest because of COVID-19, because of the, you know, as I mentioned, endoscopy has been quite limited. So we've been doing some of this implementation a bit quicker, perhaps, than we'd thought a year ago. Then there's the question about whether we do need other big trials. Do we need to show that this improves mortality outcomes? So, you know, is it enough to show that we detect more cases of Barrett's and we detect earlier stage cancer, or do we actually need to show that we stop people dying from cancer of the esophagus? And actually those sort of trials need to be very big, and you need follow-up. And been having discussions just this morning about whether that was something that's necessary, and different ways of doing it and designing it. And some of this research you can actually do while you're implementing technologies. So there are nice designs you can do so that you can actually be using it in clinical practice at the same time as still collecting research outcomes. And I think that's something the NHS is getting much better at doing. And that's really ideal because people start benefiting from the technology, but you're still evaluating it all the time. So I think that's something we're really keen to do. So in terms of the next big things, I want to see if we can really start doing some implementation. Let's continue to evaluate it while we're doing it, and see if we can diagnose the other main type of cancer of the esophagus for use, particularly in poorer parts of the world.
I think one of the next questions to finish on is that someone's asked, looking forward in 10 years, what would your dream be for the Cytosponge? I think you've touched on some of the elements there, but is it possible to even look 10 years ahead of where it could be? Is it better to say where will you be in 3 years, 5 years?
Well, who knows? I mean, I guess I'd be very pleased if this kind of technology, either Cytosponge or something very like it, is actually improving how we diagnose esophageal cancer, that we're able to do more early detection, improve outcomes, and that it's all, you know, improved the management of this, the detection and management of this disease. That would be fantastic. And you know, it's been very interesting and exciting to see that since we've gone on this journey, there are a lot of people who are now thinking about similar sorts of approaches. They're developing their own devices, their own biomarker tests. You get competition in the field, which is actually a great thing. And so whether it's Cytosponge or something like it, it will be very satisfying if it all comes to something and it actually improves patients' lives. And that's, I think, really what matters for me.
It's been great. Thank you so much, Rebecca, for your time, and thank you to everybody who's joined us over this evening. It's been incredibly useful to see how the processes that you've gone through and the challenges that you've had. Coming up, there will be more CBC on virtual tour events. In October, we have Richard Gilbertson from the CI Cambridge CRUK Cambridge Institute coming to talk to us. We also have Billy Boyle from Owlstone in November, and we'll be having a talk on cystic fibrosis in December. And there'll be other things that will be coming up in 2021 as we go forward. So just like to say thank you again, Rebecca, for your time. Thank you to Jill Wilson in the background for all the help she's done, and to be informed about all the other events that we've got coming forward. If you just check out our webs, the Biomedical Campus and this Cambridge University Health Partners website, all the information will be there. So hope everybody has a good evening, and see you all soon. Thank you.
Thank you. Bye-bye.